Vice-Chancellor and Principal of the University of Pretoria, Professor Tawana Kupe, Professor Themba Musia, Vice Principal, Student Life at the University of Pretoria, members of the University of Pretoria Executive, Professor Marleen Kock, Lecturer and Medical Scientist in the Department of Medical Microbiology in the Faculty of Health Sciences, members of the Faculty Executive, including our school chairs, colleagues, students, and friends of the faculty, distinguished guests. I am Tiaan de Jager, Dean of the Faculty of Health Sciences at the University of Pretoria. This evening's inaugural address will be presented by Professor Marleen Kock and will focus on the 21st century antimicrobial resistance. Where are we and where to next? Please allow me to welcome Professor Themba Musia, Vice Principal, Student Life at the University of Pretoria, to introduce Professor Marleen Kock. Professor Themba Musia has taught at schools and tertiary institutions and occupied prominent positions in academia over the past 3 decades. His current substantive position is Vice Principal, Student Life at the University of Pretoria. This portfolio includes the strategic management of all extracurricular student development services, health and well-being support, oversight of Tux FM, as well as student life and student housing. His academic training is in politics, governance, and quality assurance in medical education, management, law, and he holds a PhD in policy studies, particularly focusing on governance and transformation in higher education. Professor Themba Musia.
Thank you, Professor de Jager, Dean of the Faculty of Health Sciences. And good evening, ladies and gentlemen. Welcome to this special occasion. I hope you will enjoy this evening with us as Prof. Kock keeps us captivated and as she delivers her inaugural address. On behalf of the Senate of the University of Pretoria, I would like to make use of this opportunity to extend a special word of welcome to Professor Kock herself, my colleagues in the executive who may be present here, the Dean of the Faculty of Health Sciences, Professor Tiaan de Jager, his Deputy Deans, Professor Steenkamp, Professor Mashamba Thompson, and Professor Sinkubuke, and the faculty management team. Other members of senior management of the university, colleagues in the Department of Medical Microbiology, the Faculty of Health Sciences, and those further afield who have taken this time to share in this wonderful occasion. Members of Prof. Kock's family across the country, it's a big family, who are proudly watching and who have provided all the available support to her throughout the years to build her career. And I would like to single out Prof. Kock's mother, Vina Kock, her brothers, Henry, Petrus, and Jürgen Kock, her sisters, Sulen Bosman, Jacomine Visachi, as well as her aunt, Jane Stolper. And last but not least, our wonderful students, who we serve so much with energy as they make us proud to be Turkeys. We are pleased that you can share this event with us, ladies and gentlemen. There are a number of auspicious occasions in the life of an academic. These are typically marked by distinctive university rite, ritual, and traditions. One of those occasions is the customary inaugural address, which takes place upon an academic's promotion to full professorship or appointment to a position of academic leadership. The inaugural address provides a forum for the university to showcase its role models and to celebrate the endeavors that really matter in academia. The special significance of the traditions in a university lies in the measure to which they reinforce values and importance of a good education, a strong work ethic, and our contribution to society. Celebrating excellence in this way is part of our proud culture and heritage as an institution of higher learning. This evening, we therefore get to celebrate the exciting academic ventures in which Prof. Marleen Kock is involved. Other academics will have an opportunity to sit back outside their daily pressures and their own disciplines and listen to their respected colleague with undivided attention and even gain some new perspective as we navigate these inter- and transdisciplinary areas of research. Those who sit on the outside of academia, like family and friends, will be able to get a sneak peek into Prof. Kock's passion and the results of the years of hard work. And last but not least, Prof. Kock will have our undivided attention as she shares with the audience in her world a portion of which excites her. But first, for those who don't know Professor Kock personally, I would like to share some important background. Marleen obtained a BSc cum laude, BSc honors cum laude, MSc cum laude, and PhD in the Department of Microbiology and Plant Pathology at the University of Pretoria. In her PhD studies, she focused on identifying and characterizing nitrogen-fixing root nodulating bacteria of honeybush tea. After completing her PhD in 2004, she was able to apply the knowledge and expertise she had gained by working as a research fellow at the SAPI Technology Center in South Africa. The project in which she was involved described the bacteria responsible for paper breaks in paper mills. In 2006, she was appointed as a postdoctoral fellow in the Department of Medical Microbiology at UP, which gave her the opportunity to shift her focus and interest in disease-causing pathogens, their genetic relatedness, and antimicrobial resistance mechanisms. Then in 2007, Prof. Kock was appointed as a lecturer, medical scientist, in the Department of Medical Microbiology at UP, as of course part of the NHLS, and her identification, her special interest in bacterial taxonomy formed the basis of her research, focused on the molecular epidemiology and antimicrobial resistance mechanisms of clinically relevant nosocomial and community-acquired gram-negative pathogens. During her time as an academic, she published 56 research articles. She saw 46 BSc honors, 35 master's degree, and 12 PhD candidates to successful completion of their postgraduate degrees under her supervision or co-supervision. She acts as a reviewer for several ISI-related journals and is an experienced examiner of postgraduate research output, and she was recently invited to write a review article on Acinetobacter baumannii. This is a difficult scientific, I'm not a scientist, for clinical microbiology reviews, an opportunity and challenge that she is ready to face. In her inaugural address entitled 21st Century Antimicrobial Resistance, Where Are We and Where to Next? She will share research into measures to address increased antibiotic resistance and virulence causing outbreaks in South African healthcare settings. Over the past 2 years, the outbreak of COVID-19 pandemic has drawn global attention to the impact of infectious diseases on human health, social and economic well-being. Our first-hand experience during this time has made us realize anew how crucial it is to understand the origin, characteristics, and effective prevention of control measures of infectious diseases. But while COVID-19 rages on, she will touch on another silent pandemic that poses significant risks to global public health. This pandemic is the emergence of the spread of multi-drug, extensively drug and pan-drug resistant pathogens, pathogenic bacteria belong to the escape pathogens. It is indeed a complex and exciting field, and the research and expansion of knowledge in this regard, and is a major scientific, medical, and societal significance. At the risk of bringing Russians and Americans together, given the tensions we experience in Ukraine, a Russian-born American 20th century writer and philosopher, Ayn Rand, wrote that live and act within the limit of your knowledge and keep expanding it to the limit of your life. And Prof. Kock, may you continue to expand your knowledge and add to the research that impacts on our lives positively. But let me not take much of your time. Ladies and gentlemen, I present to you our esteemed colleague, Professor Marleen de Kock, and we look forward to her address. And thank you.
Good evening to my esteemed colleagues, my family, friends, and students. Thank you for joining me tonight for this lecture. Thank you, Prof. Musia and Prof. De Jager, for being here to support me. This is a brief outline of my presentation. I hope at the end of the lecture you will understand why I love working in this field. So briefly, I will have a background, discuss infectious diseases, antimicrobial resistance, antibiotics, bacterial antimicrobial resistance, the molecular epidemiology of critical priority pathogens, Acinetobacter baumannii, Escherichia coli, Klebsiella pneumonia, Pseudomonas aeruginosa, and then end off with some important points to remember, key strategies, and my acknowledgements. So what is microbiology? Microbiology is the study of very small things that we cannot see with the naked eye. It has always been a question to uncover which agent is the cause of food spoilage or a specific disease. Ancient civilizations already understood that spoilage and disease is caused by something unseen. It was attributed to bad air, or the spontaneous generation theory, or perhaps something called germs. Greeks attributed disease to bad air. In Roman times, there was a very clear separation of human waste with complex sewage system and water sources, the aqueducts, were made. Several prominent researchers and clinicians made important contributions to microbiology and medicine. Hippocrates recognized the link between unseen agent and disease. Antony van Leeuwenhoek, a Dutch cloth merchant and scientist, made the first microscopes to study water samples and made drawings of that. Ignaz Semmelweis, a Hungarian gynecologist, recommended hand washing before and after performing a clinical examination. Louis Pasteur was a French chemist, and he was known as one of the key figures in the golden age of microbiology. He developed the first vaccine against anthrax, developed pasteurization, something that we are all familiar with, and also the key principles of industrial microbiology. Robert Koch was a German clinician who developed Koch's postulates, which are the key steps to prove that a specific pathogen causes a specific disease. Joseph Lister advocated the use of phenol in surgical procedures, which was the birth of aseptic surgery. Salman Waxman discovered streptomycin, which is produced by specific soil bacteria, and that also led to a whole spectrum of different discoveries for different antibiotics. Joshua Lederberg received the Nobel Prize for his description of the process referred to as conjugation, used by bacteria to share genetic material. These images highlight the differences between a prokaryote and a eukaryote. Prokaryotes have a very simple cell structure. They do not have a nucleus or any specialized cell structures. Microorganisms include bacteria, and we refer to them as prokaryotes. Then we have the yeast, molds, together the fungi, protozoa and helminths, grouped as the parasites, and these are all eukaryotes. Viruses are included as a microbe, but they are not living. They are acellular. They are not living cells. A virus consists of proteins and the genomic material, which can be either RNA or DNA, and eventually it takes over the host cell, uses the cell's machinery to produce progeny, and ultimately kills the cell. Bacteria are microscopic, simple single-cell living organisms. There are two groups of bacteria, namely the bacteria, true bacteria, and those are all the bacteria that we encounter in our daily life. The archaea, the ancient bacteria, survive and grow in extreme environments, which can be hot, cold, high pressure, or even chemically challenging environments. Important enzymes used in biotechnology industry and in diagnostic and research laboratories originate from these archaea. To prove that scientists have a sense of humor and a dramatic flair, one of the important lineages of the tree of life is named Asgard, and one of the smaller branches include Loki archaeota. Bacteria are everywhere, or almost. The environment selects. Some examples include hydrothermal vents, soil, plants, water, Arctic ice, animals, hydrothermal springs. It is very important to note that most of the bacteria cannot be cultured in the laboratory. It's only through the advances in technology that we are able to study the bacteria around us by using culture-independent methods. Microbes make an important and positive contribution to our lives. We use microbes in different industries. Microbes can use different nutrients or carbon sources, follow different metabolic pathways, which result in the formation of different end products. Examples include food and beverage and animal feed production, chemical production, bioremediation, especially when there are oil spills, and in the case of acid mine drainage. In the case of biotechnology, manufacturing of insulin and other enzymes used in the paper manufacturing industry, plant growth promoting stimulants, photosynthesis, most of the oxygen on Earth is made by microbes, and then biological water treatment. This image highlights the nitrogen cycle and the important role bacteria plays in maintaining a balanced nitrogen cycle. This is also a flashback to my PhD research project where I focused on root nodulating bacteria of honeybush tea plants. Of course, there's also a dark side to microbes. The negative impact of microbes on our daily life has become very clear during the current COVID-19 pandemic. These are only a few examples where microbes go over to the dark side, and it includes food spoilage, economic loss due to the spread of microbial diseases in forestry, crops and fruit trees, paper breaks in paper mills due to microbial growth, formation, which is when a community consisting of different microbes establish on a specific surface and then leads to the breaks in water distribution systems, morbidity and mortality linked to infectious diseases in animals and humans. The environment, plants, animals, humans, all have a microbiome, which is very unique to that particular niche. The human microbiome is unique to the specific site. Studies have shown that it plays a very important role in immunity, metabolism, and maintaining a healthy microbial balance. Several factors play a role in the composition of the microbiome. In the case of the gut microbiome, it is influenced by diet and antibiotics, and to a lesser extent, host genetics. The normal microbiome of a site can change as a result of factors like antibiotics or hormones, which then lead to a dysbiosis of the microbiome. This is a shift in the microbial population from being beneficial to overgrowth of harmful microbes, which affect the host health negatively. Before the time of COVID-19, each year there were 2.5 million new HIV infections, 300 to 500 million new malaria infections, and 8 million new TB infections. This led to an estimated 5 million deaths per year. In the case of sexually transmitted infections, especially the 4 main curable pathogens, they account for 1 million, mostly asymptomatic, which means we don't see any signs or symptoms, infections per day, leading to 354 million new infections of either gonorrhea, chlamydia, syphilis, or trichomonas per year. STIs increase the risk for HIV acquisition and transmission. Having an STI impact on reproductive and sexual health, also having an impact on pregnancy outcomes. We know that the COVID-19 pandemic and associated lockdowns had a very negative impact on the primary healthcare setting. Patients did not attend clinics, medication was not always available, patients defaulted on their treatment, etc. When we look at the leading causes of death in low middle income countries like South Africa, 4 out of the 10 leading causes of death are linked to infectious diseases. Note the position of neonatal conditions, which include infections, lower respiratory infections, diarrheal diseases, and tuberculosis in terms of deaths per year. In South Africa, we are now in the midst of a typhoid outbreak caused by Salmonella typhi, which is a known water and foodborne pathogen. It is endemic in South Africa. Carriers and infected people excrete the pathogen, contaminate water sources. People become infected when using the water for drinking or washing of food or coming into contact with someone that has typhi. The picture in low-income countries is even darker in terms of deaths due to infectious diseases. 6 of the 10 causes of death are linked to infectious diseases. Malaria and HIV are the 2 additional causes. Globally, the impact of infectious diseases are further compounded by antimicrobial resistance. A very recent Lancet publication used published data and models to estimate the global impact and contribution of antimicrobial resistance to deaths. The authors focused on hospital-acquired infections and reported that 4.95 million deaths were associated with antimicrobial resistance in 2019. This included 1.27 million deaths, which could be directly attributed to AMR bacteria. So what is antimicrobial resistance? Microorganisms can become resistant to antimicrobial agents, and that includes antibiotics, antifungals, antivirals, and antiparasitic agents. Anything used to treat or prevent infections in animals, plants, and humans. And some worrying examples include malaria. increased reports of parasite resistance to antiparasitic drugs used, like the artemisinin-based treatment, are seen. And then also the malaria vector is resistant to the insecticides used. HIV evolves quickly and has resistance to several antiviral agents used against the virus. A recent study reported the emergence of a more resistant and virulent HIV circulating in the Netherlands. Candida auris is an invasive pathogen causing bloodstream infections in ICU patients and patients requiring invasive devices, and is resistant to several antifungal agents. Tuberculosis, there are several reports, and we know they are resistant to rifampicin and isoniazid, which are the first-line antibiotics, also resistance to second-line, and some of the newer antibiotics have been recorded. In the case of gonorrhea, one of the 4 curable STIs, it's the increased reports of resistance to several antibiotics, so it's multi-drug resistance, penicillin, quinolones, azithromycin, tetracycline, and there are also reports of resistance developing against the last-line drugs used. So what is the impact of antimicrobial resistance? It leads to increased morbidity or suffering and then death. It is a very significant impact on the economy and healthcare system of a country due to prolonged hospital stays, increased pressure on staff, increased diagnostic tests, and also more costly treatment, and then of course the patient is not at work. So how does it spread? Microorganisms adapt and change to survive in the changing environment. Resistant microorganisms are found in animals, the environment, food, plants, and humans. It can spread from person to person, and also from person to animal, and also via food. So what drives the spread of antimicrobial resistance? The lack of clean and safe water for animals and humans. Antimicrobials are often misused or overused. We see fake drugs, some people that like to prescribe antibiotics, and also easy access to antibiotics, and people not finishing their antibiotics. So several factors. Poor infection prevention and control strategies in hospitals and settings. Gaps in the knowledge and lack of awareness in the general public. Increased use of disinfectants and antibacterial agents in homes and personal care. Increased occurrence of resistant bacterial populations in the community setting, environment, and animals. And limited access to affordable medicine, diagnostics, and vaccines. And then, of course, legislation is not enforced. So where does antibiotics come from? So we have the antibiotics, penicillin, coming from molds. There, Alexander Fleming discovered the effect of a mold, Penicillium notatum, growing on a plate of Staphylococcus bacteria. He saw that it inhibits the growth, and that led to the discovery of penicillin. The case of Salman Waxman discovered streptomycin. There's also vancomycin, erythromycin, chloramphenicol, all of it coming from Streptomyces. Then some antibiotics are semi-synthetic, so it's adjusted, improved on the naturally occurring antibiotics. And then the other antibiotics are totally synthetic. Examples there include linezolid and metronidazole, which is one of the drugs used against parasites. So one of the new drugs, cefdoricol, is like a Trojan horse. It looks like a special complex used by bacteria, so it's familiar to them, and therefore they deem it harmless and then transport it into the cell. And once it is in the cell, it targets the cell wall structure of the bacteria. So there are some important definitions when you look at antibiotics. It can be bactericidal, so the antibiotic kills all the bacteria. Bacteriostatic, the antibiotic inhibits the growth of the bacteria. So it does not kill the bacteria, all the bacteria. the immune system of the host should clear the infection. Then we also look at the antibiotics have different spectrums. So some antibiotics have a narrow spectrum. The antibiotic is only active against a few bacterial species or genera. Broad spectrum, and there they have a big spectrum of activity against gram-positive and gram-negative bacteria, so diverse group of bacteria. So if we focus on bacterial antimicrobial resistance, how does bacteria become resistant? So this can happen during a process of selective pressure, so chromosomal mutations can arise. Here the genetic code is changed, which leads to changes in the target of the antibiotic. And then the most concerning ways in the clinical field of antimicrobial resistance development are acquired through either the uptake of naked DNA from the environment, that process we refer to as transformation. One of our pathogens, Neisseria gonorrhea, the pathogen linked to gonorrhea, is an example that has all the receptors and necessary proteins to take up foreign DNA from the environment and then incorporate it into their genome. Then we see the mobile genetic elements, which are a major concern in hospital-acquired pathogens. So these are extrachromosomal genetic material which can spread between bacteria, move to and on a chromosome, copy and move to other bacteria. And there are several different antimicrobial resistance determinants which can be transferred between bacterial pathogens. Then there's also phages, a genome that can insert into a chromosome. So what are the mechanisms of antibiotic resistance? It can be non-enzyme mediated, which just means the antibiotic cannot enter the cell, or it's actively removed from the cell once it enters, or the antibiotic can no longer bind to the target because it doesn't recognize the target anymore. Or it can be enzyme mediated. In this case, the antibiotic inactivates. The antibiotic is inactivated by the bacteria. So it's important to remember that bacteria often have more than one mechanism of resistance. Once an antibiotic becomes available, resistance often develops quite quickly. It is important to remember that non-pathogenic bacteria harbor antimicrobial resistant determinants as a way to protect them against the antimicrobial products produced by other microbes in the environment. Therefore, antibiotics increases the selection pressure on the bacterial population to either evolve or to die. Some groups or clones of a specific bacterial species are better adapted to survive and spread. We refer to these as superbugs. So these pathogens are multi-drug resistant, which is they are resistant to at least 3 classes of antibiotics, or extensively drug resistant when they are resistant to all antibiotics except the 1 or 2 of the last available antibiotics, and then pan-drug resistance when they are resistant to all available antibiotics. The critical priority pathogens were identified by the World Health Organization, and they include multi-drug resistant bacteria causing infections in hospitals, nursing homes, and also patients on ventilators or catheters. And these include Acinetobacter baumannii, Pseudomonas aeruginosa, and then the Enterobacterialis, which includes Escherichia coli and Klebsiella pneumonia. These bacteria are often resistant to carbapenems, and that's the best treatment available for these multi-drug resistant bacteria. And these bacteria form part of the escape pathogens as they escape treatment and cause serious infections. So my field of interest is to describe the molecular epidemiology of critical priority pathogens, which is the characterization of bacterial isolates using a combination of phenotypic and genotypic methods to ultimately determine the characteristics, So looking at the resistance profile, enzyme production, also relatedness, and then looking at evolution of the different isolates, their resistance genes, virulence genes, and how they spread in an environment. Everyone knows that a PCR test is used for COVID diagnosis. I will quickly explain how PCR works, as it's something we often use in diagnostic and research laboratories. The method ensures that we can amplify a specific region of interest to a level that can be detected. So short fragments of DNA are designed to bind to an area of interest. And with the help of a special enzyme, the first one originated from archaea, and the enzyme structure is special as it remains stable at high temperatures. So the area of interest is then copied several times, and after each cycle of different temperatures, there's an exponential increase in the number of the specific gene products. The products of this process can either be detected by visualization of the products using special chemical dyes that bind to the double-stranded DNA and fluoresce under ultraviolet light, or newer technology as that used in COVID testing, which allows the detection in real time by using instruments with light sources that excite fluorescent dyes, which then emits at a specific wavelength that is captured and read by sensors. Software programs then capture the results and display it on the screen. The other important method frequently used in clinical and science laboratories is Sanger sequencing, which is based on the same principle as PCR, except that each reaction includes only one special short fragment to start the process. It also includes the normal bases and special fluorescently labeled base pairs, the adenine, thymine, cytosine, guanine, which then allows extension of the fragments once one of these is added, ultimately leading to fragments differing by one base pair. These fragments are then separated based on size. Each stop base has a different fluorescent label attached to each, which is then read by the instrument and recorded. The next big advancement in science was the development of next-generation technologies, which generates massively parallel sequencing data, is high throughput, generating millions of reads per run, which can be used to determine the sequence of exons, and that is your coding regions of eukaryotic chromosomes, targeted sequencing, which is targeting then specific regions of the genome, and whole genome sequencing of a specific organism. We all know how important this method is in the surveillance of SARS coronavirus 2, and how it has assisted in uncovering the hidden mysteries of this virus. In the case of bacteria, the genome is extracted, cut into fragments, special labels are added, it's amplified. One fragment is then immobilized in a well and amplified and then analyzed. One of the key elements of next-generation sequencing is its ability to identify variants or changes in the genome. So the reads are assembled against a reference genome, and that is a genome that we know contains accurate and correct results. So changes are then identified as single nucleotide polymorphisms on the sequence genome. And now we have third-generation sequencing, which generates sequence data of the complete genome of a microbe without having to fragment it. And nowadays, we can use a hybrid approach with a short-read data as well as the long-read data to generate good sequence data. So if we now look at the different organisms, Acinetobacter baumannii is an environmental bacterium. It inhabits the skin and mucous membranes, survives for very long periods on both dry and moist surfaces, which is already already a problem in hospitals. It's got intrinsic, adaptive, and acquired antimicrobial resistance. It caused large outbreaks in several healthcare settings globally, and patients at risk include people who have weakened immune systems, patients on a ventilator, or any patient with an invasive device, like, for instance, urinary catheters. Prolonged hospital stays, also those in intensive care units, patients with open wounds, and it can spread by person-to-person contact or then contact with contaminated surfaces. We also see community-associated pneumonia, especially in tropical climates, and more at-risk patients are then your alcoholics, smokers, and diabetic patients. So in a study done in our department, Acinetobacter baumannii isolates were collected from public laboratories serving teaching and district hospitals, and that was 2013-2014. And we identified important antimicrobial resistance genes in these isolates. And these genes can spread from resistant to susceptible isolates. Several multi-drug resistant clones, meaning they are resistant to at least three different classes of antibiotics, were established within hospital wards. Some of these clones were also present in the other hospitals, and this highlights the spread of clones via the transfer of patients and also the movement of staff. So the presence of antimicrobial resistant clones poses an increased risk to hospitalized patients. So currently we have also looked at carbapenem-resistant clinical Acinetobacter baumannii isolates collected from a public laboratory and then also a private laboratory. And what we saw is that now all the isolates were extensively drug resistant. So that means only last line antibiotics remain. And some of these isolates, so 11 already of the 141, were already pan-drug resistant, which means there are no available antibiotics left. So all these isolates harbored several antimicrobial resistance genes. Whole genome sequencing results revealed the presence of several different beta-lactamases. So it included oxyacylinases, different ones, also ESBL genes, and I want to just highlight Acinetobacter-derived cephalosporinase and then New Delhi metallobeta-lactamase. So the ADC is induced when the patient is treated by using beta-lactam antibiotics. NDM is a very important antimicrobial resistance gene. It leads to resistance to all the beta-lactam antibiotics and also beta-lactamase inhibitors. And also it carries lots of other resistance mechanisms. And that is exactly what we saw. So the several co-resistance genes, also to antibiotics, metals, and then disinfectants. Genetic relatedness study showed that there were several of these highly resistant clones in the hospitals across South Africa, because we looked at several of these isolates. And they were basically limited to no antibiotics available for treatment. And it highlights the importance of strict adherence to and implementation of infection prevention and control strategies. In a new project, we will be elucidating the dominance clones evolution over time, looking at isolates before and those taken off during the time of COVID-19. So Escherichia coli is a normal inhabitant of the gastrointestinal tract, and it can cause disease in humans and in animals. And there are different pathotypes that are identified based on the disease they cause. So we have the extraintestinal pathogenic Escherichia coli, the XPEC, and they can be classified even further into uropathogenic E. coli, sepsis-associated pathogenic E. coli, and neonatal meningitis-causing E. coli, and also the avian pathogenic E. coli. Then that's another groups, the intestinal pathogenic E. coli, and those are linked to gastrointestinal infections. So there's a very important clone that we are worried about. The pandemic clone is Escherichia coli ST131. This clone is associated with multi-drug resistant, and again, so free drugs is out, and then it's also virulent. And it is found in the community setting. So, and it's one of my MSc students study. He looked at non-repeat Escherichia coli isolates, and those were collected over a 1-week period in October 2019. And it was from a private diagnostic laboratory, so these originated throughout the country. Phenotypic and molecular tests were performed, and 2/3 of these isolates were from outpatients, and these images, so the majority of the ST131 isolates were from outpatients. The multi-drug resistant clone is present in the community, it can spread in the community, and the antimicrobial resistance genes are mobile, so it can be shared with other bacteria. We also found an emerging or other high-risk clones, ST69 and 73, and both these clones are frequent causes of UTIs and associated with antibiotic resistance. One of my current PhD students has detected ST131 as a causative agent of almost 40% of bloodstream infections that are caused by E. coli in both the public and private healthcare setting. And then another PhD student did a One Health study in Zimbabwe, and this study was part of an AMR surveillance study conducted by the National Reference Laboratory. And we looked at the molecular epidemiology of XPEC isolates circulating in humans, animals, and the environment in Zimbabwe, looking at using phenotypic and genomic analysis, and that was performed by Mr. Takavira. So the MDR isolates in humans, animals, and the environment was seen. They were resistant E. coli clones that are established in the hospitals and community, and there were several clinically relevant antimicrobial resistance genes identified in poultry and also the environment. And the antimicrobial resistance genes included extended spectrum beta-lactamase genes, and these are all also coded on mobile genetic elements so they can spread. Also mobile genetic elements coding for quinolone resistance, even colistin resistance. And this is all a potential public health threat because these isolates can act as a reservoir of ESBL positive E. coli and antibiotic resistance genes. It poses an increased risk to users of contaminated water, and it also highlights the need for access to sanitation and safe water. So whole genome sequencing of 48 selected ESBL positive isolates from urine specimens were done. So it included representatives of different geographical regions and years of isolation. And what we saw is that predominantly sequence type ST131, and then another important high-risk clone, ST410, were the most important antimicrobial resistant clones and responsible for the increase in extended spectrum beta-lactamase producing Escherichia coli strains since 2017. So we saw that mobile genetic elements, the ones identified, lead to an increase in bacterial fitness. It enhances the acquisition of resistance and virulence genes. And then we also found some new ST131 strains, or just an evolved clone already. And that was identified during the period of 2017 to 2018, showing the establishment and evolution of this antimicrobial resistant ESBL clone in Zimbabwe, posing an important public health threat. The figure on the left shows the Zimbabwean UTI isolates, their year of isolation, the specific clone, its antimicrobial resistance genes, virulence genes, and relatedness in terms of changes over time. The figure on the right shows the evolutionary relationship of the Zimbabwean pandemic clone S3131 isolates compared to those from other African countries, as well as other parts of the world. The AMR genes and the type of the specific extrachromosomal genetic material or plasmid is also indicated. Klebsiella pneumonia. So Klebsiella is normally found in the gut of animals, humans, also in the environment. And the problem with Klebsiella pneumonia is that it acquired antimicrobial resistance genes, and it includes resistance to extended spectrum beta-lactamases, carbapenemases, etc. In the healthcare setting, it is linked to pneumonia, bloodstream infections, urinary tract infections, wound, surgical site infections, and meningitis. It can also cause community-acquired infections, causing lung and liver abscesses and serious eye infections. And then we see hypervirulent strains, and that's what is linked to the community-acquired infection. These are normally not antibiotic resistant. Now there's a convergence of hypervirulence and antibiotic resistance. So strains that are previously just hypervirulent can acquire antibiotic resistance genes, and some of the hypervirulence traits can also be transferred to antibiotic resistant clones. So we characterize carbapenem resistant isolates collected over several years, and with the help of our local and international collaborators from Canada and the US, we described the emergence and spread of a high-risk clone causing outbreaks in private hospitals. The high-risk clone was linked to antibiotic resistance, and it is referred to as ST307. This clone, in our setting, was associated with special enzymes, and in this case, which hydrolyzes carbapenems, and this enzyme is oxycylinase 181, and it is also on plasmids, and these plasmids are known troublemakers, the INK-X3 plasmids. These plasmids are often associated with New Delhi metallo-beta-lactamases and carry several other resistance mechanisms on the same plasmid. The ST307 clone accepts several carbapenemases, including NDM, Klebsiella pneumonia, carbapenemases, OXA48-like Verona integron-mediated metallo-beta-lactamases, and all these mobile genetic elements can be shared. This figure on the left highlights how the ST307 clone with OXA181 emerged and then spread within a very short period of time across South Africa. And you'll see, if you look at the colors, different colors. And the figure on the right, my apologies, it is very busy. But the very important point is to note that the South African isolates belong to a very special and separate clade. In summary, the study showed the rapid spread of the high-risk clone across 6 provinces, 23 cities, and 42 hospitals. And it affected 350 patients. It highlights intra-hospital, inter-hospital, intercity, and inter-province movements of patients. ST307 emerged in the middle 1990s in USA, and then it spread globally, causing nosocomial outbreaks. It always harbors other resistance mechanisms, and this clone seems to be very fit, and it persists in an environment. And it looks like it is set to globally replace the major Klebsiella pneumonia clone So the study highlighted the importance of molecular surveillance for tracking emerging antimicrobial clones. And one of the ongoing studies is elucidating the clonal identity of the isolates harboring more than one carbapenemase gene. So is ST307 circulating in the community? Carbapenem-resistant isolates from community-acquired UTIs and rectal carriage isolates were obtained from a private laboratory in Gauteng. And the rapid screening assay developed in the previous study was used to detect ST307 and to determine the presence of the specific plasmid and also the flanking regions of the OXA gene. So what we saw is that 26% of the isolates from the community-acquired UTIs were ST307, and then ST307 in 10% of the fecal carriage isolates. Spread of AMR is very real in the community. So the fecal carriage shows that these patients are at an increased risk of developing carbapenem-resistant infections. It's very important for the cohorting of patients to avoid transmission to other patients and strict adherence to infection prevention and control measures. So Pseudomonas aeruginosa, the last pathogen, are ubiquitous bacteria found in soil, water, plants, animals, and surfaces. It grows in water and requires minimal nutritional substances, so we can find it everywhere. Its maximum growth temperature is 42 degrees Celsius. It's a very opportunistic pathogen, and it's very well adapted. So it can use different metabolic pathways, and it likes resistance. So healthcare-associated infections can lead to 5% to 17%, and that is linked to Pseudomonas aeruginosa. And they can be healthcare-associated and ventilator-associated pneumonia, burn wound infection, surgical site infection, urinary tract infection, and bloodstream infection. So what we see with multi-drug resistant and extremely drug-resistant infections is that they are linked to a spread of high-risk clones, defined as epidemic clones that exhibit then MDR or XDR phenotypes. There are community-associated infections that can also be caused by this pathogen, and then also it's very important in cystic fibrosis patients. So in a study by one of my PhDs, she looked at carbapenem-resistant Pseudomonas aeruginosa and found that very high-risk clone linked to the NDM was found in these Pseudomonas aeruginosa isolates, and that these are all present on mobile genetic elements which can be spread to other species and genera. And it's of concern that this virulent pathogen spreads in the hospital environment. So the extremely drug-resistant phenotype was identified in 54 of the isolates, and these isolates all contained one of the important carbapenemase genes. And it's again high-risk clone, and it just highlights the need for local and national surveillance. So we have to remember that microbes do not recognize international borders. So there of concern is international travel, trade, and medical tourism. Fecal carriage and spread of resistant pathogens are very real, and it leads to the establishment of reservoirs of antibiotic resistance genes. There are very limited antibiotics available for treatment. So normal surgical procedures will become complicated if we do not change our ways. And then low middle income countries will face the biggest challenge. So we have to be responsible and considerate citizens. So effective communication, training is all important to raise awareness. Surveillance and research to expand local and international data is very important. Using a One Health approach to manage and avert the crisis. And then capacity development is critical, infrastructure investment is also critical, and we have to implement sanitation, hygiene, and infection prevention measures to prevent the spread of infection, and then the appropriate use of antimicrobials in the human and animal health. So there must be an investment in the development of new antimicrobial agents, quick and accurate diagnostic tools, the use of artificial intelligence to predict phenotypes, and also how antimicrobial pathogens will spread, screening assays, new vaccines, especially for vulnerable populations, and also other interventions like bacteriophage treatment, immune modulators, or antivirulence modulators. So my acknowledgements, I would just like to thank University of Pretoria and National Health Laboratory Service for giving me the opportunity to do what I love, and also for their funding, also the National Research Foundation, my Dean, Prof. De Jager, then my mentors, my colleague, Prof. Remco Pieters, who's also my office partner, for my Professors Lombard and Matsumura for all their help with my student, Dr. Giselle Perano for always being willing to share all her knowledge, for all my pathologists that always are willing to assist my students. Thank you. My colleagues at the department, the Pathology Institute, the faculty, and also the rest of the university. And also, of course, my amazing students, those graduated and current students. And then on a personal note, I will not be here, I wouldn't have been here without, by God's grace. Then my parents, my brothers and sisters, my extended family, my friends, I thank you.
Ladies and gentlemen, please allow me to thank Professor Marleen Kock for her insightful inaugural address. Professor Kock's address highlighted the impact of infectious diseases on human health, social and economic well-being, which we have all experienced and lived through due to the current COVID-19 pandemic. There are many lessons that we have gained throughout the pandemic. One key lesson is the importance of understanding the origin, characteristics, and effective prevention and control measures of disease. Today's inaugural lecture brought forth awareness about emergence and the spread of multi-drug, extensive drug, and pan-drug resistance pathogenic bacteria. The identification of an emerging highly successful clone by Professor Kock linked to antibiotic resistance and virulence causing outbreaks in South Africa's healthcare setting is commendable. I trust that today's inaugural lecture will inspire academics to be proactive in protecting society from outbreaks, and that many will be encouraged to step up into the future by incorporating new technologies to help with effective infection control and prevention strategies. Colleagues, let's continue to conduct research that matters and make a difference locally and globally. Thank you, Professor Kock, for your contribution to the pool of knowledge on this important topic and for being a hashtag life changer. Finally, allow me to thank Professor Themba Musia for leading us today in this inaugural lecture and for being a friend of the Faculty of Health Sciences. members of the University of Pretoria Executive, for your continued support, Faculty Executive Committee and Marketing and Communication Office at the Faculty of Health Sciences, the team at Future Africa, and Louis Klute Productions, thank you for making this inaugural address possible. Distinguished guests, staff, students, and audience, thank you for joining us this evening. Stay safe, remember to vaccinate, and good night.
Gaudiamus igitur juvenes dum sumus. Gaudiamus igitur juvenes dum sumus. Post jucundam juventutem, post molestam senectutem, nos habet